Welcome, everyone. This is the Masterclass. My name is Patrick Nazareth. I have around 30 years of experience in teaching, and I'd like to help dispel your doubts, should you have any. Today, we shall be looking at the chapter The Making of a Scientist, written by Robert W. Peterson. Like, subscribe, and share. Robert W. Peterson was an American journalist and author who wrote extensively about science, sports, and achievement for young readers. He has contributed to various educational publications. He is known for making complex scientific ideas easily accessible and inspiring to young students. He has a biographical writing style, combining factual accuracy with engaging. He focuses on the human element beyond scientific achievement. He profiles exceptional young minds and the factors that contribute to scientific greatness. How often have we been told to work hard, to study harder? There was an article in a very important journal, The Proceedings of the National Academy of Science. It told us about a new theory on the working of cells, published at the age of 22 by a person named Richard Ebright. It was the first time such an important scientific journal had published the work of a college student. You could compare it to the way Tendulkar or even Virat Kohli went around the world breaking records. For Richard Ebright, it was just the first in a long list of achievements that he got in science and other fields. What did he start with? It started with butterflies. Richard was an only child, no siblings to play with, therefore football, baseball out of the question. He immersed himself in reading, collecting things, whatever he could lay his hands on: stones, fossils, butterflies, coins. He even became an astronomer, peering at the stars at night. Two things helped him. He had a lively curiosity, and his mother encouraged his interest in everything, buying him all the equipment that he required, taking him on trips, and helping him in many other ways, just like a mother does. His mother says that she was his only companion until he started school, and then she began to bring friends home to him. His father died when he was in the third grade, and that left only the two of them alone at nights. Every evening round the dining table, they used to work, discuss things together, and his mother kept on giving him new things to learn. Richard Ebright soaked up everything like a sponge. By the time he was in his second grade, he had collected all 25 species of butterflies that were in his hometown. That would have been the end of it, but his mother gave him a book, The Travels of Monarch X. This opened his mind to the world of the monarch butterfly. At the end of the book, there was a suggestion that readers who were interested in butterflies could write to Dr. Frederick A. Urquhart of Toronto University in Canada. His mother did. Urquhart responded, sending them tags that they could use to put on butterflies, and the tag butterflies could be studied by him once they were captured by others. How do you go about tagging a butterfly then? Butterflies have a short shelf life, 6 weeks or so. If you simply try to collect butterflies in those 6 weeks of summer, you would be running out of butterflies very soon. Richard went a step further. He caught a female butterfly, collected her eggs, watched the eggs grow to caterpillar, to pupae, to butterfly, and then tagged them. It was a tedious job, but he stuck at it for several years. But he himself says only 2 butterflies that he tagged were recaptured. In the 7th grade, he went to a science fair and entered. You would expect him to win, but he lost. Why was that? Because his experiment was very sterile. He simply put slides of rock tissues. Other people performed real experiments. He learned from his mistake. To prepare for the next year, he wrote to Dr. Urquhart, "What can I do?" And Dr. Urquhart sent him a list of experiments that he could try. He tried to find the cause of a viral disease that killed all monarch butterflies every few years. He thought it came from a beetle, this fatal disease. It didn't. But this experiment of his that disproved the beetle theory went on to win the prize. The next year, he tried to understand why another type of butterfly, known as the viceroys, copy how the monarch butterflies look. He thought to himself that maybe it is because monarch butterflies do not taste good to birds, and therefore viceroys try to become like monarchs because they taste good to birds. So a bird looking at a viceroy butterfly disguised as a monarch will not eat it. He kept starlings as pets. He fed them bird food. Then he offered them monarch butterflies, and he noticed that they left the bird food and went for the monarch butterflies. This led him to understand that his theory was wrong. Again, this particular experiment led to him being placed first in the zoology division and third overall at the science fair. Richard was now in high school. In his second year, he tried to find the answer to something very simple. There were gold spots on the pupa of the monarch butterfly. Why was that, he wondered. Perhaps it contained a hormone that was necessary for the butterfly's development. He devised an experiment to show that this was true. And this project won Ebright first place in the county fair. That gave him an entry into the International Science and Engineering Fair. There, this project took the third place for zoology. This gave him a chance to work at the Army Institute of Research, the Walter Reed. As a high school junior, He continued his experiments on the monarch pupa. That year, his project won first place at the International Science Fair, and he got another chance to work at the Army Institute. In his senior year, he went a step further. He extracted the hormone that was there in the gold spots, and he showed clearly how monarch cells, if not provided with that hormone, do not grow into the butterfly scales that we associate with the monarch, whereas the ones that do get the hormone develop into a beautiful monarch wing. That project won the first prize for zoology at the International Fair. After graduation, he continued working at the Army Laboratory, and this time he had even access to the laboratory at the US Department of Agriculture. He was now in Harvard in his freshman year, and using the advanced machines at the laboratory of the Department of Agriculture, he was able to isolate the chemical structure of the hormone. It was here, a year and a half later, that he got his theory in the journal. It was a new theory about cell life by looking at X-rays of the chemical structure of a hormone. His roommate, James Wong, and him together, they work all the night. What are they doing? Making drawings, constructing models of this new theory, which will soon be published in the journal with them both as co-authors. But why is this so important? Because how does a cell read a blueprint? We all carry DNA. The DNA is the blueprint of life. But how are we able to replicate it so exactly? This provided the answer. Richard graduated from Harvard, second in class, and continued there, becoming a graduate student researcher, continued with his experiments to test his theory. Why is this theory important? Because if proved, it could be a great step towards correcting cancer and other diseases. All this starting where? Just because of his scientific curiosity. The spots of a monarch pupa leading him to the theory of cell life. Richard Ebright has always been interested in science ever since he began collecting butterflies. But he's not one-dimensional. He found time to become a champion debater, to become a person who loves the outdoors. He's an expert canoeist. He's a very good outdoor photographer as well. He's also very good at public speaking. He was a high school straight A student. And because he found learning so easy, his energy turned towards debating and model United Nations clubs. He found someone to admire here, Richard A. Wehrer. It was mutual. His teacher was happy that he had found somebody who would not just work on his class projects but still find the time to work on debates. Not because he wanted to win, but because he wanted to present the best possible side that he could. And if you try to become or present the best, you usually win. So, the author tells us these ingredients form the makings of a scientist. First, you have to have a first-class analytical mind. You have to add curiosity. You mix in the will to win for the right reasons. Not because you have to win, but because you want to be the best. From the time the book, The Travels of Monarch X, Richard Ebright has tried to open wider, look deeper into the world of science, because he's always curious to find or look for something new. Who are the characters in the story then? Richard H. Ebright, an exceptionally bright student, only child who grows up in Pennsylvania. He begins collecting butterflies in kindergarten. He's deeply curious, competitive for the right reasons, a straight A student, a champion debater, and a public speaker. Ebright's mother, a devoted single parent after her husband's death. She only worries about her son, his primary companion, his greatest supporter, spends evenings helping him learn, finds educational activities for him to do. Dr. Frederick Urquhart. He's a professor at the University of Toronto. He studies monarch butterfly migrations. He involves Ebright in his scientific experiments. He sends him experimental suggestions. Richard A. Wehrer. He's a high school social studies teacher. He is into debating. He opens Ebert's mind to new ideas. James Wong, Ebert's roommate at Harvard University, who's the co-author of the paper which explains the cell theory. Wong works all night helping Ebert develop molecular models. What are the settings of this particular lesson then? It is the mid to late 20th century. It spans the time Ebert is in kindergarten to graduate school. What places are mentioned? Reading, Pennsylvania, a small town where Ebert grows up. His basement, where he starts to conduct his first experiments. County science fairs, where Ebert wins prizes upon prizes and understands what real science experiments are. International Science and Engineering Fair, where he tastes global recognition. The Walter Reed Army Institute, where he works in the entomological laboratory. Harvard University, where he develops his cell theory and graduates with the highest honors. Harvard Medical School, where he continues research as a graduate research student. What are the literary devices that the author gives us? Firstly, analogy. Publishing in a scientific journal is compared to hitting a home run. Chronological narrative. The story follows the author from kindergarten to the time he graduates. Direct speech in quotation. Extensive use of Ebright's own words and those of his mother makes the account feel more personal, more authentic. Metaphor. DNA is described as the blueprint for life. The loss at science fair as a sad feeling. Contrast. Simple display, losing versus real experiments, winning. Competitive spirit for right reasons versus winning for prizes. Imagery. Stack of suggestions from Dr. Urquhart. 12 tiny gold spots on a monarch pupa. Working all night drawing pictures and constructing plastic models. Understatement. Surprising no one who knew him, Richard Ebright graduated with highest honors. What are the themes covered in this chapter? Curiosity as the foundation of science. Genuine curiosity drives Ebright's entire journey. Role of encouragement. His mother's support is crucial to his development. Learning from failure. Losing at the science fair teaches him what real science is. Winning for the right reasons. Ebert competes to do his best, not just to win prizes. Holistic development. True greatness involves multiple interests besides one speciality. That's the lesson there for you then. I do hope I have been of help. If I have, please do remember to like, subscribe and share. Remember, there would be others who may require our help. Thank you so much for watching.